Welcome to Alpha Biochem videos, and in this video we're going to discuss glycolysis. Now, I'm going to do my best to explain this to you very simply and very easily. At first, when you look at this reaction step, it looks very complicated, but it's very easy once you get your head around it. And the main difficult thing to remember is just the positioning of or the order of each reactant and product, and also the enzymes used. But once you go over it a few times, it does become very easy. So I'm going to go over the main steps, and by the way, if anybody has any questions, just write them in the comments below. So let's begin now. Firstly, of course, since this is glycolysis, we're going to be discussing glucose. So firstly, we have a glucose molecule here, and what this, well, to start the reaction, it gets converted into glucose 6-phosphate. Now this reaction is catalyzed by an enzyme called hexokinase, and the difference between the reactants and the products is there's a phosphate group which has been added on. So that phosphate group will come from ATP, and then it will be converted into ADP. So there's one phosphate group which gets added onto the glucose. Now after this step, the glucose 6-phosphate gets converted into something called fructose 6-phosphate. So that's a fructose group with a phosphate group added onto it. Now this reaction is catalyzed by phosphoglucose isomerase. Okay, and from this reaction to fructose 6-phosphate, it's a reversible reaction, so it can occur in both directions. Now from fructose 6-phosphate, what we get is a product called fructose 1,6-biphosphate. Now what this means is there is an extra phosphate group which has been added on, so that obviously will come from ATP, which will then be converted into ADP, and the enzyme which catalyzes this reaction is something called phosphofructose kinase 1. Okay, now after the step of this reaction, there are two possible directions the reaction can go into. Now the first one is from fructose 1,6-phosphate forming glyceraldehyde 3-phosphate. Okay, now this is catalyzed by an enzyme called aldolase. So fructose 1,6-biphosphate gets converted into glyceraldehyde 3-phosphate. Now the other direction the reaction can go into is to form dihydroxy acetone phosphate, and it's also catalyzed by aldolase. However, dihydroxy acetone phosphate will get converted into glyceraldehyde 3-phosphate. So regardless, it's still gonna carry on and go towards convert into glyceraldehyde 3-phosphate, even though it's separated and went in a different direction here. So the enzyme which is going to catalyze dihydroxy acetone phosphate into glyceraldehyde 3-phosphate will be triose phosphate isomerase. Okay, now from glyceraldehyde 3-phosphate, what we get is a product called 1,3-biphospho glycerate. And this reaction is catalyzed by an enzyme called glyceraldehyde phosphate dehydrogenase. Now, something to note at this stage, the electron or the electron carriers which are used in this stage are NAD+, which gets converted into NADH, and that acts as basically an oxidizing agent. So glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate. Okay, now to go from 1,3-biphosphoglycerate, the next stage of the reaction forms 3-phosphoglycerate, and with this reaction you get one of the phosphate groups basically is removed. It says biphospho, which means there are 2 phosphate groups, but here it's just phospho, so one of the groups has gone. So obviously ADP will take up that phosphate group and get converted into ATP, and the enzyme which catalyzes this stage of the reaction is called phosphoglycerate kinase. Okay, now the next stage following this, we're nearly done now, you get from 3-phosphoglycerate to 2-phosphoglycerate. Okay, and the reaction is reversible. Also, this reaction is reversible. Okay, and the enzyme which catalyzes this stage of the reaction is called phosphoglycerate mutase. It's easy to remember because from 1,3-biphosphoglycerate to 3-phosphoglycerate it's called phosphoglycerate kinase, and from 3-phosphoglycerate to 2-phosphoglycerate the enzyme is called phosphoglycerate mutase. Okay, now from 2-phosphoglycerate, the next stage of the reaction forms phosphoenol pyruvate. Okay, now it's reversible also, and a H2O group can either be removed or can enter into this stage depending on which way the direction of the reaction is. So from this stage, from 2-phosphoglycerate to phosphoenol pyruvate, the enzyme is called enolase. Okay, and finally, to conclude these steps to glycolysis, the phosphoenol pyruvate will get converted into just pyruvate, and it's again the phospho group will be removed, so you'll have ADP, which will be converted into ATP, and this stage of the reaction is catalyzed by pyruvate kinase. So this is basically the steps to glycolysis. If you have any questions on any part of this stage or any part of this reaction, please write the comments below and I'll answer them straight away. I hope I've explained it in an easy to remember method, and like I said, make sure you comment, rate, subscribe, and thanks for watching. Bye bye.